Hello, my name is Galit Zimmerman and I'm a student at Ben-Gurion University of the Negev in Israel. Today I will talk about approximate search for known gene clusters in new genomes using PQ trees. This is joint work with Dina Svetlitsky and my MSc advisors, Mirav Zehavi and Michal Ziv Yukerson. Microbial genomes are typically organized in operons. An operon is a unit of neighboring genes that are transcribed together and translated to proteins together. Genes that are consistently located close to each other in the genome usually encode proteins that participate in the same protein complex or the same pathway. So despite extensive shuffling of microbial genomes, functionally related genes may be constrained to remain close to each other due to natural selection. Here, for example, there are two operons that are located close to each other. One operon encodes proteins of a pump that exports toxic heavy metals out of the bacterial cell. The other operon encodes a pair of proteins that regulate the transcription of the genes of the pump. A group of genes that are located close to each other in multiple genomes is called a gene cluster. A gene cluster is more general than an operon. It can represent an operon, but also a suboperon, multiple operons, or in general, a group of genes that are conserved together. Here, you can see a gene cluster of a known operon that appears in several bacterial genomes. In each genome, the gene cluster appears in a different order. These are the gene orders of the gene cluster. We would like to represent the gene cluster using some kind of data structure that will model the possible evolutionary events that yielded the various gene orders. We do this using a combinatorial data structure named PQ tree. A PQ tree is a rooted tree with three types of nodes: P nodes drawn as circles, Q nodes drawn as rectangles, and leaves. The PQ tree represents a string by reading the labels of its leaves from left to right. In fact, a PQ tree represents more than one string by allowing some reordering of its nodes. The children of a Q node can appear in either left to right or right to left order. As you can see here, by flipping the order of the children of the right Q node, we obtain a different string. The children of a P node can appear in any order. By shuffling the order of the children of the root P node, we obtain another string. Though originally PQ trees were used to represent permutations, we use an extended, more general version that represents strings. In contrast to permutations, in a string, each character can appear more than once. The PQ tree you see here was built using the gene orders of the gene cluster we saw before. You might ask yourself, why is the PQ tree a good representation for gene cluster evolution? Well, the biological assumption here is that operons evolve by progressive merging and inversion of suboperons, where the most basic suboperon is a single gene. These two types of evolutionary events are represented by the PQ tree. The first type is inversion, which conserves the order between the suboperons as they are inverted together. The conserved gene order could be related to the order in which the corresponding proteins interact in a metabolic pathway. Conserving the order means the suboperons can appear from left to right or right to left, so it is represented by a Q node. This is the case here. The genes G to M were merged to a Q node, and so did the genes C to D. The merging and inversion events happen recursively, and so after C and D were inverted, they were inverted together with their neighbor E. The second type of evolutionary event models convergent evolution, where suboperons were assembled in different species by separate events, and so they do not necessarily appear in the same order in different genomes. This is represented by a P node, because the children of a P node can appear in any order. Now, let's turn to the new problems we address in the paper. The most basic problem is PQ tree membership. Given a PQ tree and a target string, we would like to know whether the string can be derived from the tree. We would like to know if it can be obtained from the tree by ligand recursive rearrangements of the PQ tree nodes. This string cannot be derived from the PQ tree, but this one can, as the mapping lines indicate. The second problem is PQ tree alignment. In this problem, rearrangements guided by the PQ tree are allowed, as well as deletions from the tree, deletions from the string, and substitutions. Generally, for a gene cluster, there should not be many deletions, so bounds on the numbers of deletions are given as input, but they can always be set to infinity for a more general variant. PQ tree alignment is an optimization problem, and the objective is to find a string that can be derived from the PQ tree that is the most similar to the target string. Here, similarity is measured as a sequence alignment score. So why would we want to use PQ tree alignment? Well, let's suppose we are given an already known gene cluster that was discovered and studied in one genomic dataset, for example in chromosomes. And we want to identify new instances of it in a new genomic sequence that evolved independently, for example a plasmid genome. So we want our alignment model to tolerate various types of noises which are associated with annotation, sequencing, and assembly errors, and also to accommodate the fact that the target genome could have evolved independently. On the other hand, we want to be as specific as possible. So we use this grammar-guided alignment and allow rearrangements that are constrained by the PQ tree that was built from the query gene cluster. More generally, in our application, we have a long genome and the string derived from the PQ tree is much shorter. So in the paper, we actually address the PQ tree search problem. In the PQ tree search problem, we are given a PQ tree and a string representing a new genome. The characters of the string correspond to a gene orthology family alphabet. We are also given a substitution function and parameters dt and ds that bound the number of allowed deletions from the tree and string. We wish to compute the best scoring alignment of the given PQ tree versus every substring of the target genome string. There are previous works that describe efficient algorithms on PQ trees. For example, PQ tree construction, reconstruction of ancestral genomes, and physical mapping. However, we do not know of any previous work that used PQ trees for alignment or search. So here is our contribution. We defined new problems in comparative genomics, PQ tree alignment, and PQ tree search. We proved they are NP-hard, and we proved they are each fixed parameter tractable, FPT, with the parameter gamma. Here, gamma is the maximum degree of a node in the PQ tree. This means that we showed an algorithm that solves PQ tree search with a time complexity that is a reasonable polynomial times some function of gamma. In our case, the function is 2 to the gamma. Our polynomial, which is concealed here by O star, is comprised of the length of the input string n, the parameter gamma, the bounds on the number of deletions dt and ds squared, the number of p nodes in the input PQ tree, MP, and the number of q nodes in the tree, MQ. We implemented our algorithm for PQ tree search, named it PQ Finder, and made it available on GitHub. We proved that PQ tree alignment is NP-hard by showing a reduction from the job interval selection problem, JISP for short. Here it is on the left. On the right, you can see the PQ tree alignment instance resulting from the reduction. Due to lack of time, we will not get into any more details on the reduction and proof. Earlier we saw the problems defined in the paper, and now we'll dive into the algorithm itself. We'll start with a high-level overview of the algorithm for PQ tree search, which solves PQ tree alignment for every substring of the target string. It is a dynamic programming algorithm that goes over the nodes of the tree in post order, and in each step it computes the best alignments with the string. The algorithm starts by computing the possible alignments of the leaves. Here you can see that the leaf with the label D was substituted by the character U. Then, the algorithm computes the alignments of each internal node according to its type. Q mapping for Q nodes and P mapping for P nodes. Now let's continue with our example. X1 here is a Q node, so it is aligned to the string using the Q mapping algorithm. Because the children of a Q node must appear in sequential order, Q mapping is quite similar to sequence alignment. However, the children of a P node, like the node X2 here, can appear in any order, so P mapping is a more challenging task. Here are two possible alignments of X2. One is to align it to the substring UFE, and the other is to align it to the substring FED. We store both options. In fact, we store the best alignment for each substring of the input string versus each subtree of the PQ tree. So now, when we move on to align the root of the PQ tree, there are also two optional alignments. Either align the whole tree to the substring from A to E, or delete the character U and align the whole tree to the entire string. Next, I will explain the P-node mapping algorithm in more detail. I will skip the explanation of the Q-node mapping algorithm because it is simpler. As we saw in the previous slide, the algorithm is given the optimal alignment of each child of the target P-node versus each substring of the input string. We can think of every alignment as a colored interval on the real line, where each integer corresponds to an index of the input string, and the color of the interval represents the aligned node. This interval here, for example, corresponds to this alignment, which is an alignment between the blue node X2 to the substring of the input string between the indices 3 and 4. Thinking about every alignment as an interval with the color of its node means the objective is to choose a set of non-overlapping intervals where each color is chosen at most once. The intervals you see here are the intervals from the previous slide drawn horizontally. The P-node mapping algorithm we suggest is inspired by the algorithm that solves the job interval selection problem. However, JISP does not consider uncovered sections of the real line, which in our case means deletions from the string. In addition, our problem differs from JISP by the fact that we return an optimal solution for every substring of the input string. Our algorithm is a dynamic programming algorithm, and at each step we consider a different prefix of the real line and look for a colorful set of known overlapping intervals that covers it. Due to lack of time, I will only explain how to reduce the problem instance to a smaller one by shifting the endpoint at each step. Based on this understanding, it is possible to formulate a recursion rule and design a dynamic programming algorithm. We have here the intervals from the previous slide, and at the moment we consider the prefix that has the endpoint 9. To make sure we choose intervals that do not overlap, our aim is to choose at most one interval at each endpoint. So for a specific endpoint, we either delete a character from the string and reduce the endpoint by 1, or we choose an interval that ends at the endpoint and reduce the endpoint accordingly. Here, we can either choose the dotted green interval or the dashed blue interval. Let's continue with the last option as an example. Because we already chose the blue interval, we cannot choose any more blue intervals. We also cannot choose the rightmost green interval and pink interval because they are beyond our new endpoint. Intervals that we will not be able to choose are marked here in lighter colors. Once more, we can either delete a character or choose an interval. As an example, let's say we chose the pink interval. Our new endpoint is 3, and we mark that we use the color pink. There is no interval that ends at index 3, so we can only delete a character. Now we can choose the dotted green interval, move the endpoint, and reach the end. In this example, by choosing these 3 intervals and deleting one character, we covered the section from 1 to 9 of the real line. So in the derivation of the PQ tree that has the best alignment to the prefix of the input string that ends at index 9, the order of the children of x is x3, x1, and then x2. Here, we used all the colors, which means all the children of x were mapped, but in general, we can delete some of them. Now, we move on to the biological results section. A typical microbial cell contains one chromosome and some plasmids. Plasmids are small DNA molecules that can replicate independently from the chromosome. While chromosomes carry essential genes, plasmids usually carry genes that are beneficial for the cells of the host in specific conditions, for example, in antibiotic stress. Plasmids are considered a major driving force in microbial evolution, providing mutation supply and constructing new operons with novel functions. The fact that environmental stress can lead to rearrangement of the plasmid genes, which in turn can lead to an adaptation to new stressful environments, gave us the motivation to search for chromosomal gene clusters that are rearranged in plasmids. In our analysis, we extracted gene clusters from chromosomes of about 1,500 genomes, and then constructed picture trees based on their different gene orders. Using the picture trees as queries, we applied picture tree search to find instances of the gene cluster in plasmids. We had two objectives. The first was to find tree-guided rearrangements in plasmids. These are gene orders in plasmids that are represented by the picture tree but did not appear in the chromosomes. The second objective was to explore the different variants of a known gene cluster. In this case, we allowed each variant to differ from the original gene cluster by substitutions, insertions, and deletions, in addition to the possible gene rearrangements. Using picture tree search, we obtained 29 gene clusters that had tree-guided rearrangements in plasmids. Here you can see the top 10 gene clusters sorted by the specificity of their picture tree. The third top-ranking result is a gene cluster encoding a heavy metal efflux pump and the two-component system that regulates it. A heavy metal efflux pump is a protein complex that is interleaved in the membrane of the bacterial cell. It is involved in the resistance of bacteria to a wide range of ions of toxic metals. This pump was found in 7 plasmids, where one plasmid contained a tree-guided rearrangement that was not present in the chromosomes. Interestingly, this tree-guided rearrangement was found in the bacterial strain that was isolated from an environment polluted with several heavy metals. To achieve our second objective, we searched for approximate instances of the gene cluster of the heavy metal efflux pump that possibly encode related variations of the pump. The picture tree of this gene cluster was used as a query and reran against all the plasmids in our dataset. This time, in order to increase sensitivity, deletions, insertions, and substitutions were allowed. Exact instances of the query were detected in plasmids, as well as 3 variants. One variant had a gene deletion, another variant had an insertion of a gene, and the last variant had a substitution of a gene by another gene. The exact instance of the query was significantly enriched in beta-protobacteria, and the variant that had a gene insertion was significantly enriched in gamma-protobacteria. The variant with a gene insertion encodes a heavy metal pump that is specific to copper export. The inserted gene encodes a copper chaperone that supplies copper to the efflux pump. This variant was found in the plasmids of 6 genomes, from which 5 genomes are of known pathogens. This is sort of surprising, so we ask the question, why do pathogens need a copper efflux pump? Well, previous studies suggest that the host immune system exploits excess copper to poison invading pathogens. This can explain why these pathogens evolved the copper efflux pump that serves as a bacterial defense mechanism. In conclusion, we defined new problems in comparative genomics, denoted picture tree alignment and picture tree search, and we designed an efficient algorithm that solves them. We showed that picture tree alignment and search are NP-hard and that they are both FPT with the parameter gamma, the maximum degree in the tree. We implemented the algorithm as a tool and made it publicly available on GitHub, along with all the data needed to reconstruct our biological results. We found 29 chromosomal gene clusters that had tree-guided rearrangements in plasmids. One of the tree-guided rearrangements could be the result of a heavy metal stress. a variant of the heavy metal Efflux pump that included an insertion of a gene, revealed that Efflux pump found mainly in pathogenic bacteria, and it is a possible bacterial defense mechanism against the host immune system. A future extension of this study could explore whether tree-guided rearrangement events are correlated with environmental stress or other bacterial adaptations. I thank my co-authors and all the people and agencies who supported this research, and thank you for your attention.